Today we are going to be talking about the cell theory. What is a cell? The cell is the smallest living unit inside living things. And the cell is divided into two particular categories, which are unicellular and multicellular. Unicellular is basically when a living thing is made up of only one single cell. Multicellular, on the other hand, is the complete opposite. It is basically when living things are made up of multiple cells. Who discovered the cell and when was it discovered? The cell was discovered by an English scientist, Robert Hooke, 1665, who actually studied cork, which is the dead cells of an oak bark. When he was studying the cork, he realized or noticed the repeated geometrical box-shaped structures, and he decided to call them the cell. The cork was dead, so he was actually only studying a cell wall and an empty space, not an actual cell. What is the cell theory? Cell theory is summarized into three main points. First one is that all living things are made up of cells, which we all know. And the second thing is that cells are the basic units of structure and function in any living thing or organism. And the third thing is that new cells are basically produced from pre-existing cells in the living organism. Who was the first person to discover the microscope? The microscope was discovered by Anton van Leeuwenhoek. He was born 1623 and died 1723. He was the person who perfected the simple microscope. He created the first microscope known, 1674, by using the tiny lenses that magnify 300 times. The lenses that he used to create the first microscope, he used them to examine blood, yeast, insects, and other tiny objects that he could find. He was the person who discovered the microscopic world. Now we're going to talk about two specific kinds of microscopes or types. First type is the dark field microscope. The dark field microscope is used in a dark field where the object that is on the microscope lens is bright with a dark background. This is made possible to see by a special kind of dark field condenser that is put in the microscope. This dark field microscope is used to identify the living unstainable cells and the thin bacteria-like spirochetes that cannot be visualized by any light microscope. Second type of microscope is the phase contrast microscope. It is used to visualize the living cells by creating a small difference in contrast between the cell and water. It converts a slight difference in the refractive index in cell diversity, which is not easily detected by the cell does not easily detect the variation in light intensity. This microscope is useful for microbial mobility and finding out the shapes of the cells and the bacterial components. Now we're going to talk about the two types of cells. First type is the eukaryotic cells. Now, eukaryotic cells are basically cells that contain a nucleus and the organelles, and they are closed by this plasma membrane. Organisms that have the eukaryotic cell are specific. They are the protozoa, the fungi, the plants and animals, and obviously the human body. These organisms are grouped in a biological domain that is called the eukaryota. What is the eukaryotic evolution? The evolution of cells is stated by the hypothesis that eukaryotic cells evolved from a symbiotic association of prokaryotics by the endosymbiotic theory, which is particularly well supported by the studies of the mitochondria and the chloroplasts, which are thought to have evolved from a bacteria living in a large cell. What are the prokaryotic cells? Prokaryotic cells are cells that do not have a true nucleus or membrane-bound organelles. The organisms within a domain, bacteria and archaea have the prokaryotic cells, but any other forms of life have eukaryotic cells. And the prokaryotic cells are shaped into capsules. And last but not least, what is the cell diversity? Cell diversity refers to a range of cell types that compromise a tissue or an organism. It arises during the development of the nervous system as a result of acts of the cell intrinsic and extrinsic controlling of the fate of the cell determination. I hope you enjoyed listening to my project,